Welcome back to MitoGenesis, where we uncover the tiniest engines of your cells, hold the secrets to health, healing, and longevity. Today, we enter the frontier of biological rejuvenation. You've all heard the term anti aging, but here we are talking about actual reversal of cellular age using advanced regenerative science. What if we could reset an old cell, not just make it function better, but turn back the biological clock? This is no longer science fiction. Let's begin with an iconic study. In 2016, Belmonte Group at Salk Institute showed that partial reprogramming of cells using Yamanaka factors, those master regulators of pluripotency, could reverse signs of aging in mouse model of progeria. Not to full embryonic stem cells, mind you, but just enough to rejuvenate tissues, improve organic organ function, and extend lifespan. Similarly, David Sinclair's lab has demonstrated that periodic activation of these factors in retina could restore vision in aging mice, again not replacing cells but revitalizing them. How does this tie to mitochondria? Here's the twist. Aging isn't just about damaged DNA, it's about dysfunctional mitochondria. And it turns out that epigenetic aging and mitochondrial decline go hand in hand. When we reprogram cells, even partially, we see surge in mitochondrial biogenesis, improved ATP output, and reduction in oxidative stress. Now to dive deeper into how we refine, personalize, and optimize three rejuvenation strategies, especially with AI, we are joined by a young but a leading voice in aging longevity research, Basudha Banerjee, who's completed her master's, has been doing research in various institutes, and is soon headed to Europe for her PhD. But I really love her perspective and the diversity of knowledge at this young age. Welcome, Basudha Banerjee, to this talk.
Thank you very much, Dr. Khurana. It is my pleasure to be here. And as you mentioned, you are absolutely right. Aging is increasingly being understood as a convergence of epigenetic drift, metabolic dysfunction, and a mitochondrial decay. And now we are starting to piece together how to reverse that. So let's start with mitophagy, that is the cellular recycling of damaged mitochondria. As we age, the process becomes sluggish. Stem cells, in particular, would accumulate dysfunctional mitochondria, leading to a lot of exhaustion. And now we are learning how to modulate mitophagy pharmacologically using compounds like urolithin or activating pathways like AMPK and sirtuins with NAD plus boosters. These interventions would restore not just the mitochondrial health, but most importantly, the stem cell vitality. And as you mentioned previously, partial reprogramming. Partial reprogramming is a game changer. But here's the challenge to it. Too much of reprogramming, and then we risk loss of identity or even cancer, because there is always a chance of tumorigenesis. Too little and there's no benefit. That is where AI enters the picture.
Yes, this feels like Holy Grail, finding the Goldilocks zone for rejuvenation.
Exactly. So with the multiple AI guided models, we can now simulate how cells respond to different doses and time windows of Yamanaka factor expression. So we are building predictive frameworks that factor in epigenetic age, metabolic state, and even mitochondrial dynamics, down to the ROS levels, the crystal structure, and even ATP flux. Some groups are combining single-cell multiomics with deep learning to anticipate whether a stem cell would revert, rejuvenate, or senescence based on a set of interventions. And importantly, that these models that are available, they aren't just theoretical. We are entering an era of personalized mitochondrial therapy where AI tells us, firstly, how and when to apply the NAD plus or AMPK activators. Secondly, how often to trigger mitophagy. And finally, whether a specific cell population is reprogrammable or needs full replacement.
Well, what excites me most is the synergy, epigenome restoration, enabling mitochondrial repair, and vice versa. I've seen data showing that reset in the epigenetic clock can restore mitochondrial coupling efficiency. Less ROS, better ATP, improved autophagy. It seems like system-wide tune-up.
Absolutely. And when we talk about longevity, this integrated approach matters a lot because you cannot fix the nucleus and ignore the mitochondria. They talk to each other constantly, and aging happens when that conversation breaks down. And now, with tools like CRISPR Cas9, epigenetic editing, and AI guided reprogramming, we are learning how to restore that dialogue gently, precisely, and effectively.
Aging might still be inevitable, but we are learning how to slow it down and in some cases reverse its tracks. From stem cell revival to epigenetic research to mitotropic therapies, guided by machine learning, we are scripting new chapters in human biology. Thank you for joining us on this exhilarating journey. In the next episode, we'll explore how intracellular mitochondrial transfer, yes, one cell donating its healthy mitochondria to another, is being used in therapies for stroke, cardiac arrest, and even cancer. Until then, stay curious, stay mitotropic. This is MitoGenesis signing off as Sukant. With me is Basudha Banerjee. Thank you very much.
Thank you so much, Dr. Khurana. This was indeed a very insightful episode. Thank you.